Hello students, this is experiment number 8, study of different water samples for pH, clarity and presence of microorganisms. First write the objective or the aim of the experiment, that is collect water from two different water bodies around you and study them for pH, clarity and presence of any living organism. For doing the experiment we need following materials. We need water samples. Take pond water, canal water, river water or tap water. We need pH paper, microscope, slides, cover slips, dropper, filter paper, torch, beakers and a cardboard box. Come to the theory part. Water may contain small quantities of suspended particles, organic substances and number of microorganisms. All these make the water turbid and the water is unfit for consumption, or you can say water is polluted. pH of water also vary. If pH is less than 7, then water is acidic. If it is more than 7, then water is basic or alkaline in nature. In both these cases, water is unfit for use. But if pH is 7, then water is neutral and is fit for drinking purposes. Procedure. First write procedure to test pH of water sample. Here take water samples in test tubes as shown in figure number 1. Then dip small piece of pH paper in each sample of water. You will see change in color of pH paper. Match this color of pH paper with color scale given in the color booklet as shown in figure number 2, and then note the value of pH of each water sample. Next, write the procedure for testing the clarity of water sample. In this, take a cardboard and prepare a Tyndall setup as shown in figure. Check the figure for Tyndall setup. We have to make two holes on opposite faces of the box. Then place the beaker containing sample of water inside the cardboard. Throw the torchlight from one hole and observe the water sample from the other side. Now compare the turbidity of different water samples. This is the procedure involved to check the presence of microorganisms. Take a clean dry slide. Put a drop of water separately on different slides from different water samples. Put the cover slip on water drop and remove extra water with the help of filter paper. Finally, observe the slide under microscope. You can note all these steps in the figure given below. These are the diagrams you have to draw. Draw these diagrams on left side of your practical notebook. Use sharp pencil to draw them. You can also use pencil colors to show green and blue color of the pH paper. This is the observation table. Write whatever you have noted for pH test, clarity and presence of microorganisms for both samples of water A and B. Then write the results. Sample A. The pH of sample A water is 7.0 as it changes the pH paper color to green. Thus, water sample A is neutral. Clarity. It is clear water. No suspended particles are seen in this water sample. Then microorganisms. Also, no microorganisms are seen in this sample of water. Sample B. pH of sample B water is 8.0 as it changes the color of pH paper to blue. Thus, water sample B is alkaline or basic in nature. Clarity. It is turbid water as suspended particles are seen in this sample of water. Microorganisms. Microorganisms like bacteria, protozoa, diatoms, algae, etc. are seen when observed under microscope. So, what is the conclusion? Presence of suspended particles and large number of microorganisms in sample B indicates that water is polluted and unfit for use, while sample A is clean water. Now precautions to be taken. First, the hole of the cardboard box for Tyndall setup should not be large. Second, the light source should be of sufficient intensity. Third, take clean and dry test tubes to test pH value. 4. Dry the pH paper before comparing the color with color scale. 5. To check presence of microorganisms, shake the water well before putting the drops of water on the slide. And 6th, the slide should be clean and dry before putting drops of water sample. So that's all for today. Thank you very much for staying with me. Subscribe the channel to get the information about my new video. Thank you so much.